All right. First off, I want to say happy Thursday and welcome to CRISPR Office Hours. Really excited to have you guys on today, as well as our guest. So as we get started here, I want to make sure to talk real quick about a few housekeeping items. We're going to be recording the webinar, and this will be live on YouTube, hosted on YouTube afterwards. And so today we're really excited as we're continuing CRISPR Office Hours. Last week was our first episode of season 2, as we like to call it. And you can find a recap of season 1 on our blog, synthego.com/blog, if you want to read all the guests that were on and read about what are the key topics they covered. So for us, CRISPR Office Hours was started as a way where we felt we could help the community come together, ask questions about how to navigate through these difficult times and tackle the problems we have together as a community. So for us, it's really critical that we get to interact with you during these CRISPR Office Hours, that you ask questions, that you give feedback, input, comments, as that provides more information for everyone in the community, as well as learning together how we can move forward and tackle this pandemic together. So my name is Aditya Vempati. I'm the VP of Marketing at Synthego. And as always, I'm really excited to do CRISPR Office Hours with my co-host, Kevin Holden.
Hey, Aditya. Hi, everybody, and welcome to CRISPR Office Hours, Season 2, Episode 2. So I'm your co-host, Kevin Holden. I'm Head of Science at Synthego, and the title for Office Hours this week is Fighting COVID-19 with CRISPR-Chip-Powered Diagnostics. All right, so as we've talked about a few times over the past few weeks, now's never been a better time to keep calm and carry on. As you'll recall, this poster was originally made by the British government during World War II as part of a public assurance campaign to remind people that even in the face of danger, it's important not to be paralyzed by fear, but to stay collected, calm, and to get on with what you need to do. So we think this slogan has never been more appropriate than now, as we all do our part during this time. And from us at Synthego, we'd like you to remind all of you and our audience and all the genome engineers out there to keep calm and CRISPR on. And Aditya, do you want to tell people how people can get one of these cool shirts?
That's right, Kevin. We've been wanting to make sure people do keep calm and CRISPR on. So as a result of that, we're actually going to be giving every one of the attendees today a free CRISPR Keep Calm and CRISPR On shirt from Synthego. At the end of the webinar / office hours, we'll have the URL where you can fill in information to get one. So with that, let's kick off today and talk about who we have on the panel. Obviously, we have my co-host, Kevin Holden. But we're really excited today to have Kiana Aran. And she is currently assistant professor at the Keck Graduate Institute in Claremont, California, and co-founder and chief science officer of Cardea Bio.
Hi, everyone.
So thanks for being here today, Kiana. As I'm sure you're aware, we like to kick things off with a little poll question to get everyone engaged, to get everyone interacting, and break the ice a little. So every week we pick a fun work from home / behavioral thing that people are doing during the pandemic. And this week, we want to know, what are you really doing during your Zoom calls? Are you paying attention? Are you checking out your coworker's apartment? Are you catching up on emails? Or are you live tweeting during the pandemic? Or forwarding TikToks, and my personal favorite, working out? So Kevin, what are you doing during your Zoom calls?
I guess I would say I'm trying to pay attention most of the time, but it can get pretty easy to get distracted, especially when you have a 30-minute Zoom call for what maybe used to be like a 5-minute chat with somebody. So honestly, though, it's kind of fun to check out where people live. So I'm going to go with number 2.
All right. What about you, Kiana?
You put me on the spot here, and I know that some of my team members are on this Zoom call right now. But for me, So every time you see, I'm talking with my team members who are see them on the Zoom now. Every time you see my Zoom video is off or my Teams video is off, I'm probably working out or doing some sort of Pilates exercises. And this is why I turn off my Zoom video calls. So I will go with number 6.
Oh, wow. That's a good one. For me, I think I'm definitely number 2, checking out my coworker's apartment, and probably 5, forwarding TikToks, although I use Instagram more. But from our audience, I'm seeing a lot of 1 and 3, which is paying attention and catching up on emails. And there's another one where someone said 2, also paraphrase it with kids and pups. That's a good one. And these are pretty interesting. I think the other one was checking out horses for sale online from Garrett. That definitely got my attention. So all clever answers. Thank you to the attendees for chiming in. And as always, want to make sure this is interactive. So please put your questions in the chat window and we will definitely try to get to them. So with that being said, we've been following a certain model that we've been tracking through this pandemic. And I want to make sure I give a moment to cover that. Kevin, do you want to touch a little bit on this?
Yeah, certainly. So for the past couple of weeks, maybe over a month now, as we enter day 999 of quarantine and shutdown, we've been discussing this model that was developed by the Linus Group, where they surveyed over 2,000 scientists about their thoughts and activities during the pandemic. And so depending on where we are in the world right now, I'd say most of us are probably going through this transition phase. So we know, last week we talked to somebody, a scientist, Imran House at Peter Mac Cancer Center in Melbourne, Australia. They're actually going through this transition now into the new normal. And so he was telling us about how he's been coping during this time and readjusting, right, to his new normal. And so today we're bringing in a scientist from Southern California, originally from Iran, actually, who's become quite a sensation in the last couple of years, I'd say, really bridging the gap between genome engineering, biotechnology, and kind of the high-tech world and transistor technology. So it'd be interesting to hear how she thinks she can apply the invention she's made to fighting the COVID-19 pandemic as we move into this new normal. So yeah, okay, Kiana, thanks again so much for being on the show today. And by the way, happy belated birthday to you. I know we recently discovered that you and I have the same birthday, so that was great.
Thank you. And look what I got for my birthday.
I will cut you.
Yeah, we'll CRISPR cut you. Yeah. Okay.
All right. So maybe before we jump in, Kiana, can you tell us a little bit about yourself and your current positions at both the Keck Graduate Institute and also Cardea Bio?
Sure. So I'm an electrical engineer by training, and I did my postdoc and my Ph.D. in biomedical and bioengineering. This is why I combine a lot of electronic and biology and design devices. After my postdoc, I joined Keck Graduate Institute and became a faculty there, where I also designed a lot of diagnostic devices. And in most of my diagnostic devices, I had an electrical element, biological element, because I really believe that, electronic and modern electronics should take over a lot of things in our future. About a year ago, less than a year ago, I became the co-founder and CSO of Cardea Bio, where we design, and our vision is to design the next generation of biology-gated transistor to have access to biological signals in real time. I don't know, so what else you want me to talk about? You want me to talk about how we're dealing with the situation right now, or you want me to talk about the research, or?
Yeah, maybe let's start by you telling us a little bit about the CRISPR chip technology, and also, how you had this idea to combine CRISPR with transistors.
Okay, so let me tell you about the vision first, because then it falls really nice with why I thought, combining CRISPR and transistors was a great idea. So our vision in our company is we really want to get access to biological information. The picture, if you can move to the next slide or click. I think, the picture that we have from a biology today is it's a black and white picture. It's a single snapshot of a single marker at a particular time point. For us to, to understand biology, and if you move to the next one, So we really need to understand many components and many markers at the same time. To give you an example, any change up in genomics, any change up in our gene can lead to changes in proteins expression, can lead to downstream or upstream variation in biological signals. For us to have a great understanding of underlying biological events, we really need to look at them at the same time. We look at them continuously, and by looking at one markers at a single particular time point, we won't have that picture. Imagine series of differential equations. Each variable in each of these differential equations can be a gene, can be a protein, with boundary conditions which are defined by cell membranes, by tissue type. And if you can think about it this way, the system, the biological system, can be millions of these differential equations. And we have the power to solve this differential equation. We have supercomputers that can solve this differential equations. All we need to provide, we need to feed this boundary condition. We need to feed, some relationship between these genes, these proteins, and then we can have computers solve this equation for us. So to get access to biological information, we really have to change the way we look at these biological signals. And the great thing about these biological signals, these signals are chemical, or there are physical signals that you can actually capture and look at. So the goal was, can we actually look at these signals at the same time to get a dynamic picture of what goes on in our bodies, what goes on in our systems. So if you move to the next slide. So this is where we want to be. We are the gateway between all these signals from genomics, proteomics, from other signals that you can take from biology, and we want to feed these signals to computers. We want to let computers deal with all of these huge amount of signals, because as human beings, we're not capable of processing all these signals. So where we sit in the middle, where we want to be the platform that enables this information from these biological signals to be fed into computers. And then it enables us to write machine learning algorithms and analyze those signals and get a better insight to biology. So our world is made of silicon-based transistors. Everything you have in your hand, all this gadget, all of these technological advances that you guys are using during pandemics, or these are all, were possible because of the invention of transistor. I like to call the invention of transistor one of the biggest invention of the century, which allowed us to do many, many wonderful things. So if you move to the next slide. One more. So there are many types of transistors. The transistor that we use for our applications, there are field effect transistors. I know many of you had this question of how CRISPR chip works, but then before we get to that, I want to tell you how our transistor works. So if you look at the picture on the right, what I'm showing here is two electrical terminal that you have a semiconductor material between these two electrical terminal. And the current that is passing through the semiconductor material, it's based on the conductivity of that material. However, if you apply an electric field where you put the charges, positive or negative charges, atop this semiconductor material, you can change the property, the conductivity of this material. And then we can sense these changes with our transistors. So this is basically called the field effect transistor, where you can change the electrical property of the transistor, not only by changing the material, the semiconductor material, but also changing the field. So the change in field can come from the interaction of charges with the semiconductor material. So if you have more charges, the picture on the left, you see that the current that is flowing through the system is changing, and then you can detect that in real time. So if you move to the next slide. I really like to call charges on our side, because we can really turn biological event into digital information in real time. Why? Because every biological molecule is a charged molecule, from proteins to RNA to DNA. These are all charged molecules, and they have very specific charge characteristics, and we can capture those. So it's really, the power of this field effect transistor comes from, being very sensitive to charge interaction. And because we have biology, which is also charged, we can actually utilize those for biosensing applications. So the sensitivity of our system comes from, you know, our transistor, but also we use nanotechnology, we use graphene. So between the two terminals I'm showing here, there is the honeycomb pictures that you see. This is a graphene material that we use. Why do we like to use graphene? Because graphene is a single layer of carbon molecules. It's super sensitive to charged molecules, and it's biocompatible, so it doesn't interact with your biology, it doesn't interfere with your biology. And it can transfer the signal super fast because it's a single layer, so that your signal does not dissipate like it does in silicon, where you have a bulk material. So for that reason, we use graphene. And we need to put that in perspective that graphene was invented about almost like 20 years ago. And we had so many sensors, so many publications that shows the power of this material for biosensing applications. But unfortunately, you know, you couldn't see many of these technologies out in the commercial market, and the interest started to drop down, you know, until one of the scientists who was one of the pioneers in nanotechnology decided to quit his job and really take this to the next level where you can have, you know, a commercial system for that. And so that scientist's name was Brett Goldsmith, who actually quit, you know, he did his postdoc, he published in Science and Nature and all this high impact factor journal. But at one point he got tired, so he quit his job and he started making this chips in his garage, and then he moved on to start a new startup and bring these transistors, these graphene-based transistor in massive scale. And today I'm really lucky to be able to call Brett my co-founder, and we actually both co-founded Cardea together. So this was the story of how we actually not only have this system, but we were able to actually bring those to commercial scale. So next slide. So this is where the story of combining CRISPR and graphene came to life. So I was a postdoc at UC Berkeley, you know, saw Jennifer Doudna on a daily basis, not daily basis, but most very often. So we were all exposed to CRISPR. And for me, you know, as an engineer, I was just fascinated by this technology, this little robot that can, within our cell, can unzip the double-stranded DNA, look for its target, you know, binds to its target, and then cleave it. For me, the power of CRISPR was not only gene editing, but its searching power. It was the power of it that, you know, it did not need any amplification. Within the cell, it was capable of finding its target. So the goal was like, can I actually use this search engine and monitor its activity? Can I use a search engine and combine it with nanoelectronic to see one CRISPR bind, one CRISPR cuts? So this was the whole idea, and we tested this. So at that time, I wasn't using Brett's and this commercial graphene, so I would spend months and months, you know, designing this system in Berkeley fab rooms, and out of every 10 devices, I get 1 or 2 working, and then I would run my experiment. But then I met Brett, and this facilitated the whole project, and it really not only took the project to the next level, but changed the whole way I looked at my new innovation in the lab and how I would look at them when I designed them, you know, to be more careful about not just having the next fancy invention, but also make sure that those inventions can really make an impact in a commercial world. So the way we did it, we actually put CRISPR on graphene. So we immobilized, you know, thousands of CRISPR molecules on graphene between our terminal. So the field effect transistor, you have 3 terminals, source and drain terminal, where the graphene material is sitting between the source and drain. So Aditi, if you can put your cursor on the graphene, that would be nice. Okay, and we have all these CRISPR molecules on them. We programmed this CRISPR molecule with the guide RNA, because as you know, CRISPR complex can target its target sequence by an RNA that you design for it. So this is super easy now with companies like Synthego. You just ask them, you know, to design this, and 2 weeks later you have the guide RNA in your lab. This made things super easy for me as an engineer who never worked with gene editing to actually be able to utilize the system, because, you know, guide RNA synthesis were available, you know, Cas9 Berkeley was making, Cas9, and I was able to actually use this technology. It's very fast. So immobilizing CRISPR on the graphene, and then programming it with the guide RNA that we wanted to detect, and exposing it to whole human genome, not pieces of amplified sequences. We basically assume that if CRISPR can do this in the cells, it should be able to do this on the chip as well. So this is what we did. We exposed our chip to genetic material. These are only purified genetic material without amplification to our chip, and we monitored to see if we could look at the interaction of CRISPR with the target DNA, and this is what we did. So later on, we tested the system with genetic material from patient with muscular dystrophy disorder, and we were able to detect this without any amplification. We also have a paper under review right now where we were able to detect a SNP. This is huge, and being able to detect a SNP without amplification, using not a handmade technology, but using a technology that was designed by nature, using CRISPR that was designed by nature over millions of years in bacteria, and just monitoring it was an amazing accomplishment for us. So the picture on the right shows you the chip, and if you zoom in, this is the actual chip. So the rest of it's the packaging. The actual chip is that circular part in the middle, where you can take it and put it in any sort of different configuration. So here is a video. We're zooming inside. We have 15 different transistors. This is 3-channel. We call this a triplexer. And as you zoom in, we have CRISPR compounds immobilized on the graphene material. They program with the guide RNA. And as they interact with the target molecule and they bind, we can monitor this interaction. The binding of, this is a deactivated CRISPR, so it binds, it doesn't cut, and it brings the DNA down close to the surface. And we were able to actually have sensitivity close to femtomolar with our system without amplification. And we can move. Any question till here? Because I don't want to move fast.
Kiana, actually, I have a question. How you mentioned earlier about working with your partner, Brett, to make the graphene on the chips. Has that enabled this technology that you developed, which is absolutely fascinating, by the way, to become scalable, like from manufacturing?
Absolutely. I think one of the power of our technology, and remember, CRISPR is one of the technology that was designed by nature. There's so many other molecules that we can use. I think the most enabling thing was a chip that was designed, fabricated, scaled up over many years, and this in fact enabled me to move this project, because otherwise, if I wanted to make 10 devices, have 2 working systems, and CRISPR chip wouldn't have been possible for another 5-6 years, because it would have taken you significantly more time to test those systems. So one of the major things that we have, and I think it's super important for electronic sensor, is that we have quality control of our chip. We want to make sure that we have reproducibility. We have done a lot of effort has gone into just developing the chip and making it possible to actually look at it as a true commercial sensor.
So somebody also asked a question through the chat, which is also a question I had as well, which is what kind of volumes and concentrations can you detect on the chip?
So the concentration, as I mentioned, is femtomolar of human genomics. So this is whole human genomics, and it's about 30 to 40,000 molecules right now. But again, we have not worked on significant optimization, and we are working on those right now. It's really based on your application. For example, if you're detecting genetic diseases or like genetic mutation in plants, that's not genetic mutation, but like genetic information from plants. For example, from a buccal swab, you get about 2.8 microgram of genetic material, and this is significantly more than enough for our sensor to be able to sense those things. So it depends on your application. Of course, for infectious disease, if you're looking at 10 copies, and the majority of this sensor, they're talking about, okay, we can do 10 to 100 copies. That's a bit different, and we have not gone there yet, but we are working on to see how we can not amplify the target, but amplify the signal. We haven't actually taken full advantage of the power of electronic, and we are just getting there to see what else we can get from this electronic sensor, what other information we can get. How do we amplify the signals without the need to have, without the need for the target amplification? Because the minute you start amplifying the target, then you start playing with biology, cutting it, you know, labeling it, amplifying it, and I think it just takes away from you know, the real signals that we're trying to look at.
So maybe let's talk about some applications. So
Sure.
maybe on the next slide, DJ.
Yeah. So as I mentioned, you know, we have done CRISPR, we have detected DNA, but our chip is not only a CRISPR chip. It can be used for detecting antibody antigen. It can be used to detect the small molecules, and we have collaborated with significant amount of partners that they have, each of them use the chips for different applications, for the small molecule detection, antibody antigen interaction. We have worked with CDC for detecting Zika a few years ago, where we detected the protein in the blood. So there is more. The application goes beyond just immobilizing CRISPR on our chip. So if we move to the next slide. And I think, you know, before I start this, it's really important to validate your technology through other people, because, you know, it's important that your technology works in other people's hands, because this is how you advance, this is how you improve your technology. And this is one of the strategies we're using. We like to collaborate with people. We want to see how our sensor can be utilized for their application, and how can we improve our technology for their application.
So I know you're going to talk as we go into kind of like a diagnostic application. You know, I know, you know, your company's goal and your goal has been to do biology on these chips, really, you know, to kind of enable like a new generation of computing, right? How'd you think about branching into this application?
Well, we had a lot of people approaching us for it, you know. So again, we want to be the platform company. We want to be able to provide people with a platform where they can get their information in a fast way without having to do significant sample prep and have a digital output. Because, you know, in terms of pandemics, it's super critical to have a system that can transfer the data seamlessly, you know, to a server where you can use it for tracking information. Because one of the major things that I think it's lacking in the technology today is not having the right platform to track the positive, you know, patient, you know, and it really makes biosurveillance very difficult, you know, for Google or Apple to be able to have this tracking system. You need to also have, on the other side, a good tracking system for diagnosing the patient. You know, currently, I don't know if you know about it or not, but, you know, I have talked with a lot of doctors, and it's sad to just hear that the result is still being transferred on a paper or on a PDF file. All of these new assets that are coming in the market, you know, we cannot encounter, you know, a biological threat at this scale like that with papers. So we really have to think beyond this pandemics and think about what would be next. And I think it's been recognized. A lot of people want to work with us, and this is why we thought about, okay, how can we help? Because we want to be that platform that can help people. So then we start thinking about how can our platform, you know, be useful, and what sort of a proof of principle we can provide our partner that they can utilize our sensor. So if you look at our sensor, you can do viral genomic material. You can use CRISPR technology to detect, okay, what is this virus? Is it COVID? Is it flu? And because we are transistors can be super small. We're following Moore's law, and they got smaller and smaller and smaller. So you can imagine on a small chip, you can have thousands or millions of these transistors to monitor multiple markers at the same time to increase your specificity. For example, using CRISPR, you know, you can have, for each transistor, you can have a specific CRISPR complex that targets a sequence, a specific sequence that is conserved in COVID, in flu, in other bacterial or viral infection, and not only detect, you know, to see if you have flu or if you have COVID, but do you also have another type of bacterial infection sitting atop of it? Do you need antibiotics. And, this is all, with all of these, CRISPR complexes being found and discovered, some are RNA specific, some are DNA specific. And with our technology that can provide this multiplex capability where you can detect against multiple of these analytes, proteins, RNA, DNA, it really helps, make better diagnostics, not only on a single marker, but be more specific where you're looking at multiple different RNA vectors and bacterial DNA at the same time. You can look at some cytokines. For example, if you're using serology tests, don't only look at antibodies generated against some of the surface, some of the COVID pathogens. Look at the larger number of these pathogens, because my body might interact or react to one epitope very differently than someone else's body. Let's have more of those biomarkers, and don't have just a yes or no answer. We really need to have. We really need to have a quantitative response in order to be able to guide vaccine development. How much vaccination we need? Do we need boosting? And how do we make a decision to make sure that this vaccination can provide protective immunity? So our sensor can provide that, and we have recognized that we can help our partners develop those things, and this is the whole goal. Not only that, when you look at future, this is what you need in the future. You need technologies that can transfer data seamlessly to servers where you can do all this biosurveillance more effectively.
Okay, great. So I think one of the questions that we've gotten and wanted to ask also is, there's many other, I would say, not other, but pathogen detection platforms like Detector and the Sherlock platforms that we already heard about. Can you tell us a little bit about why this is really the next generation of diagnostics platforms and some of the challenges you're expecting to face in developing it?
Sure. So I wanted to add that, we are not a diagnostic company, right? So this has to be very clear. We are a platform company. We want to be that gateway that provide diagnostic company with a platform that they can do things better, right? So I talked about the importance of looking and being specific, looking at multiple different factors at the same time. And really, my hope is we help Sherlock, we help Mammoth, we help all this, CRISPR companies to bring their wonderful assays on a digital platform, because then you can do effective biosurveillance. With a lateral flow assay, optical assay, they're wonderful to do testing at the same time. But how do you envision this to be effective in seamlessly transferring this data to biosurveillance? Unless somebody is still plugging those information in. So we really need to look at what do we need. Okay, at this time point, perfect, these assays are wonderful, they're adding values, but then for us, we're looking really beyond that. We're looking beyond what can we do for the next pandemic? How can we help with this pandemic? Providing a platform. Our platform can be a yes or no answer, can be quantitative, which could be really helpful for guiding vaccine development, because we are quantitative. We are not a yes or no answer. We can give you quantitative results, but also look beyond that. Have a vision of how do we face the next pandemic, and how do we enable all these wonderful assays that biologists are developing to be more effective in biosurveillance. So we don't want to be a diagnostic platform. We want to be that platform that help diagnostic company, bring more value to their products. Okay.
So yeah, I guess something else we've been thinking of maybe as you can move on here, Aditya. I'm thinking about how you could use this platform to make like an at-home testing kit, and why that might be important to be able to do this at home.
Right. So again, to develop something that you want to use at home, you want to have something that requires minimum user interference, right? You don't want the user to be taking pictures or analyzing their data. You want something that you put the samples and the answer is there. The importance of this, that you want something to do a daily monitoring, right? For example, if you want to monitor your health during COVID, let's say, you want to make sure that nothing happens with your pro-inflammatory cytokines level, because as this thing starts changing, you should expect something is changing in your body. Some biomarkers are changing in your body. It would be wonderful to have a system where you can test yourself, on a daily basis during pandemic, on a monthly basis, otherwise, to see how is your general health is going. And I could see that with only digital platform, because we're all getting used to all this wearable Apple Watches, Fitbit, and imagine if you can have system that can send your health information, your true health information, getting samples from your blood or from your sputum or other biological sample, and provide those at-home tests. Not only that, at-home test doesn't only require, it's not only a biological sample from our biological fluid, but also from the air that is circulating in our homes, from the sewer system, and you can get a lot of this information. And because our sensors are super reconfigurable, and this is one of the powerful things about our sensor, you take the sensor, the middle part of the sensor that I showed you, put it in any configuration that you want based on your need, and you can get use of it. So it's not, because it doesn't require, we're trying to, have this system, does not require sample handling or people pipetting in and out of stuff, and just capturing the sample, enriching it and analyzing it. You could envision a lot of different applications beyond diagnostic, for just like monitoring, environmental monitoring as well for home. So again, they're rapid. You can be multi-analyte. You can detect the small molecules. You can detect protein, biomarkers, DNA, RNA, volatile chemistries. And it's really a multiple factor sensor. You can rapidly reconfigure this. For example, if you have a system that you design for cytokine testing, you can use it for blood, you can use it for breath analysis. It's the same system, but you can put it in different configuration.
Kiana, does that mean that they're, sorry to interrupt you, does that mean that they're reusable, or are they just so inexpensive to manufacture, you could just have one for each different application?
So currently, it again depends on the application. So currently these are consumable. Of course we know how to reuse them, but if you want to have something that is FDA approved, you don't want to have a reusable system, right? And at the price point, it recently has dropped following Moore's law, and we're expecting it to drop significantly more. And it's again based on the application. So if you have an application, let's say in agriculture, and you need 1 million of this per day, that's a different price point, right? And so the answer to that is it really depends on application, the form factors that this system goes into it, and based on that, the price is designed. Also, the price is based on the demand. The price of the chip and the graphene, this is not the cost for us. The cost is the packaging. So then the packaging plays a major role based on where you're going to use the system. Are you going to use a system where the packaging is different, is more expensive, or is less expensive? Is it going to sit on a wearable system, on a plastic system? So it really depends on your need and what sort of a packaging you need for the system, and the volume. These are the things that will drive the price.
So I know a lot of you have been having questions in the chat, and thank you for being so interactive. It's great. And thank you, Michael from Cardea, for answering some questions in the chat, who works with Kiana. Maybe on that note, Kiana, since you work in both academia and industry, how are you keeping your teams in both of these places connected and inspired during this time? Maybe that also about how you got inspired to do this.
Yes, so one of the major things I have realized during the last few years is that, not everyone in academic wants to be an academic. During my training, I did not have the chance to actually be exposed to, okay, how does it look like if I work in an industry? How does it look like if I was in a management of a company? And I think it brought a different level of energy for my academic lab, just to have, both academic lab and then the innovation working like side by side, close to each other. It's been very, very useful for my academic lab. Some of these people have been hired by my company after they graduate. So it has brought a different perspective to the way my lab operates, so the way I manage the two right now, it's so first of all, during this pandemic, I actually had both teams sometimes meet with each other, because it's just, to initiate some brainstorming session. It was really good to have both team at the same time, and then we both benefited from each other. They both benefit from each other. And I don't know, it just works. It works perfect. And then the way that we have learned from being able to interact with people who were in the industry for a long time, we're actually learning how to reproduce data, how to make sure that we reduce variable in our data. And I think I have been more effective as an academic scientist being exposed to, you know, what industry needs, you know, and you know, sometimes, as you may know, reproducibility is one of the major issue in the majority of academic publication. We have learned so much about how to reduce, you know, uncertainty in our data, how to mitigate those. You know, now when we publish papers, we always have reproducibility data, which I think is super important because our goal is to take our technology from our lab to industry. This is how we leave our impact. So I love it, and I think my academic students loves it, and it really opens a different door for their future career. From my perspective, I don't know, I just love what I do. So when I wake up in the morning, I don't feel I'm working. It's just amazing on both academic and the industry side. So between being driving innovation and being able to be on the front line of something that I think will change the world, it's really great.
So Kiana,
yes.
sorry, Kiana, as we're going through, we're getting a lot of questions from the audience, so I want to make sure we have time for that. Kevin, go ahead. You're about to say something?
Oh yeah, I was just going to say, I know we skimmed over a little bit all the different potential applications. So again, thanks for doing that. We can share some of these slides and they'll be on the YouTube for you to check out later when you want to come back. So yeah, Aditya, want to take us through some questions we've had submitted?
Yeah. So question from Ava, when do you think CRISPR chip technology will be widely available?
So I need to say that, you know, we are working with many partners right now, you know, from agriculture to other industries, where we working with them to use CRISPR chip for their own applications. So in that aspect, you know, if you guys have something in mind, CRISPR chip, we are working with people to take CRISPR chip for their own applications. And you know, you can go on our website and our IPP program, where you can see how you can partner with us for your own application. On the product side, we you know, CRISPR chip, you know, we are going to have the first some of our first product out there where you can do CRISPR QC. You know, and I'm going to make sure that Synthego buys one of our robots. So you can actually monitor to see when you're using different CRISPR complexes, how are they binding to your modified guide RNA, if you're modifying your guide, if you're affecting the binding between the CRISPR, the Cas and the guide, how is your complex, you know, interacting with the DNA? You know, if you change your guide RNA, how is this interaction will be improved? You know, so we are going to have this CRISPR chip QC QC product very soon in the market as well.
Okay, great. Let me take one from the audience. Actually, there's a question here. It says, what is the size of each individual transistor, and how many binding events are needed to trigger the signal? This is from Maru.
Yeah, so so what we see, and I cannot go into detail because some of this stuff they need to be under NDA for me to be. We can play with the electronic parameter of our system to see the binding events. Very, like, I don't know how many binding events we can see, but we can track them very closely. And we are working on that next generation of chip that will enable us to do that. However, it's you know, the binding event really depends on your analyte. If you have a DNA, we've shown that, you know, within 15 minutes we have enough binding event, let's say between CRISPR and genes from Duchenne muscular dystrophy patients to be able to detect it and have a threshold above our control. So it really depends on the sample. I cannot tell you how many events because it depends on what event you're looking for. Having said that, it also can be optimized based on the amount of capture molecules or protein or CRISPR that you put on your chip, or the size of your chip. You can modulate that and you can work on that. So based on the need, we always try to work with our chip design and with our partner to design something that is suitable for their application.
So we had another question too from Maria Eugenia. What is the physicochemical basis of the activation of the transistors? And also, can you extrapolate the use of these to other proteins besides Cas?
Yes. So as I think I went through this in my presentation, where the physiochemical basis of activation of the transistor is basically the charge interaction of these biological molecules with the chip. And we monitor multiple different electrical parameters to detect those charge molecules. So basically, it's charge. It's charge interaction with an electronic bonds of the graphene, which can be detected, which changes the conductivity of the graphene. And as I mentioned, yes, our platform, it's really beyond Cas. You can use different versions of the Cas. You can use antibodies on the chip. You can use small molecules. You can use aptamers, peptides, anything that you want. We have developed those strategies to be able to use any of those capturing molecules.
Great. So we want to get here as we're running out of time. We just want to remind everyone we do have a webpage, synthego.com/coronavirus, where we have a lot of these resources listed from various publications, blogs, as well as guide sequences that we've developed internally, Synthego has, along with other publications that other scientists in the field have. Kevin, do you want to reintroduce the model here real quick, and then we can talk through the rest of the presentation?
Yeah, I think just as we wrap up, Kiana, maybe can you kind of tell us, at least from your perspective, what you've been going through, what's the new normal going to look like for you for Cardea, and also your work at the Keck Institute?
I think I will be definitely using more communications using Zoom and all this, platform. I was really against those because I like the one-to-one interaction. So definitely communication will change. And I think, we have all realized the importance of, during this pandemic, the importance of, automation and robotics, and how we really need to have more of those in our labs, in our companies, to be able to really not halt. We were not on halt, but other companies were on halt because they really needed to have people inside to run some of the experiments. So, I think it's time for us to look at automation, to look at robots more carefully. And I think this is what we're going to be looking at more.
Great. So as we're going to finish up here, Kevin, do you want to talk about who will be joining us next week?
Yeah, certainly. So actually next week, we're going to have a repeat visit. We're getting a second visit from Jonathan Gutenberg and Omar Abaduya at MIT. And they're going to talk to us about some exciting news, how their CRISPR-based COVID-19 diagnostic platform now has FDA approval. So they have the EUA, and also the new Stop COVID Diagnostic initiative they've recently launched. And that should be pretty exciting. And I think kind of also follows on, I see Kiana, you're interested in developing kind of the long-term diagnostic platform that can be, reportable and is integrated in, computer systems. And, I think in the future, it's going to be super immensely powerful. And, I think to have these guys on next week talking about what they've got that's going to help us right now with approval is going to be great too. So somebody asked if this is going to be available. Yes, it will be up on our YouTube channel later. So Aditya, do you just want to remind our audience too how they can get one of our t-shirts before we close out?
Yeah, of course. So as we said at the start, we're giving a shirt away to everybody. Keep calm and CRISPR on. You can go to synthego.com/com to get your shirt. And it's very simple. Fill out the form and put your size in, and we'll ship it to you. So first off, we want to say thank you, Kiana, for being a guest today, explaining the CRISPR-on-chip technology, talking a little bit through your experience in this pandemic and how you're dealing with it. And also for our audience, thank you for the highly interactive session. We loved all the questions. And as always, my great co-host, Kevin Holden, for making it amazing and a pleasure.
For joining us today, and Kiana, thank you again so much. It's a really interesting application of the technology, and we look forward to seeing what you're going to develop next. And we're really excited to see how it's going to help everybody. So thank you very much. And also to your team who have been online to helping us answer questions. So fantastic session.
Thank you so much for having me.
Thanks,
all right.
everybody.
Okay,
Have a good day.
everybody. Okay. Goodbye, everybody. See ya.